Okay, my name is Marie Latulippe, and I just wanted to first thank Drs. Wishart and Kadura for inviting us to talk about the project and their interest in making sure that we maintain momentum. So I am the manager of the Gut Microbiome Committee of ILSI North America, and then we'll have Dr. Paulina Piotrowski, who will describe the work that NIST is doing on the project in collaboration with the BioCollective. So if you want to go to the next slide. Just to let you know a little bit about ILSI North America, in case you aren't familiar, we are a nonprofit scientific organization that focuses on advancing specifically food safety and nutrition science for the benefit of the public. And we always like to mention that we do not lobby or advocate for specific policies. And we do our work through this tripartite model, and you see that icon to the upper right of your screen there. And in this, we bring together scientists from government, industry, and academia from both the US and from Canada. And the intellectual contribution of the scientists from these sectors is then combined with pooled industry funding, and together they decide on research questions of common interest that they would like to address. So if you go to the next slide, we have about, I think it's 9 nutrition-focused committees, and one of these is the Gut Microbiome Committee. And this committee, the one that exists now, is actually the second iteration. I think we started a little bit too early in trying to define what a healthy gut microbiome was. So we reconvened this group in 2016, and then in about 2018, the group decided to reevaluate their overall mission and focus. And so currently they're focusing on method harmonization, best practices, and standards, which is the topic of today's project, and then just generally improving the links between nutrition research and gut microbiome research. And the reason they have this focus relates to the next slide. And this slide essentially represents the mission of the committee, which is to advance the science required to substantiate dietary modulation of the gut microbiome in ways that promote health. So that sounds a bit broad, but ultimately what's needed here is a validated biomarker that represents the linkage. And this link to health is really the tricky part from the perspective of authoritative agreement. I'll give you an example. The FDA released revised guidance for what can be considered a fiber on the food label. And specific types of fibers can only be recognized on that label if they show a physiological effect, but that physiological effect also must be a benefit to health. So that link to some kind of health outcome is really critical. And the second part of this is that the evidence must be of adequate strength for a biomarker to be generally accepted. And that means that the evidence should support the link from diet all the way to health. And in order for a body of evidence to collectively point to some conclusion, it obviously has to be combined. So you look at the whole body of work and pull it together to hopefully make some strong conclusion. And thus, the committee started thinking about how to better harmonize or standardize research towards this ultimate goal. And this led us to reach out to NIST, who is the mother of all things that are standard. And they happen to be working on a human whole stool reference material for metagenomics. We decided to work together to find a way to include metabolites as part of this material. And that brings us to the event held last September. And with that, I will turn it over to Paulina to describe the project that's currently underway.
Great, thank you so much, Marie. So just to give you a little quick background, NIST is the U.S. Metrology Institute, and our mission is to promote U.S. innovation and industrial competitiveness by advancing measurement science, standards, and technology in ways that enhance economic security and improve the quality of life. And so we're very happy to be partnering with ILSI and the BioCollective in the preparation of this human gut microbiome reference material. On September 12th, the workshop that was hosted at NIST was really aimed to identify candidate metabolites that have links to diet and health that we can quantify in a whole stool reference material to provide a very comprehensive gut microbiome metabolomic product. Of course, with any reference material development, there are a number of challenges and considerations, and especially when working with whole stool, it's very inhomogeneous. There are viable bacteria present, which makes a reference material that needs to be homogeneous and stable actually quite difficult to achieve. And so all of the efforts of this workshop were recently submitted to Metabolomics. And the title of the paper you can see is The Development of a Human Whole Stool Reference Material for Metabolomics and Metagenomics Gut Microbiome Measurements. And this was really led by Rupa and Marie. Thank you so much for all of the effort that you guys have put in on this paper. We really appreciate it. On our end, we are developing the human gut microbiome reference material, and we are using human whole stool. The whole stool is being prepared by the BioCollective, which is our partner organization that's based in Denver, Colorado. They basically do all of the participant screening, recruit volunteers, collect samples, and homogenize and aliquot them. And we really hope that this reference material promotes or is adapted by the scientific community for method validation and benchmarking current protocols. As a metrology institute, we really hope that this material is a gold standard for quality control in both metagenomic and metabolomic measurements of, clinically relevant samples, but then really, any stool-based measurements that might be happening. So last summer, we participated in a pilot study where we received samples that were prepared by the BioCollective, and we performed analysis with NMR-based metabolomics and mass spec-based metabolomics, both with GCMS and LCMS, and then 16S RNA sequencing. For kind of brevity, I'm only going to tell you today about the GCMS metabolomics and some of the sequencing work that was done. The material, as I mentioned, was prepared by the BioCollective using their, what they've trademarked, the True Matrix workflow. And then to briefly describe that to you, basically, volunteers are solicited and their whole stool is collected and processed for storage. The samples are screened for HIV and hepatitis. They are homogenized with dry ice and then sublimed. And then that sublimed powder is then reconstituted in either deionized water or a buffer that's typically used for DNA stabilization. That is then filtered and then dispensed into aliquots. And so in our study, we had 5 different storage conditions that we were looking at. One of them was homogenization in water, which was then stored at 4 degrees, lyophilized and stored at 4 degrees, and then stored at -80. And then 2 of the proprietary commercial storage buffers, which are labeled PCSBA and PCSBV. And those, as I had mentioned before, are really focused on stabilizing nucleic acids. And when it comes down to the reference material, the community has really indicated to us that they would prefer intact whole cells. And so one of the trademarks of these buffers is that they lyse cells. And so right off the bat, we weren't sure that they were the kind of the right storage conditions, but because they're so prevalently used in the gut microbiome community, we went ahead and tested them anyway. And so for the metagenomic analysis, it was done by 16S RNA sequencing. I'd like to just preface this by telling you that I am a mass spec metabolomics person, and so this is a little bit out of my realm. But briefly, the samples were extracted using a commercially available kit. They were sequenced on a MiceSeq and then analyzed with a Data2 bioinformatic platform. The results that I'm going to show you today are a PCOA of the order of the microbial taxa. And one of the things that you can notice right away is the fact that the samples that were stored in water at 4 degrees are very different from all of the other preservation techniques. And that is largely what we hypothesize to be the result of bacterial growth that is occurring at samples stored in 4 degrees. So as I mentioned, fecal matter contains viable bacteria, and the BioCollective has shown that their process preserves the cells. And so at 4 degrees C, because there's a lot of other matrices in the fecal matter, sugars, lipids, fiber, etc., those conditions are can still sustain bacterial growth. And so right off the bat, we know that storing these fecal materials in the long term at 4 degrees is not feasible. When we look at some of the GCMS data, this was done by extraction using solid phase microextraction. And then, so we're only looking at the volatile metabolites. You can see that an overlay of all of the different storage conditions leads to very different profiles in the metabolites of the samples. And this is kind of concerning because we would like to have a standardized way of looking at the metabolites. And so if every preservation technique gives us a different set of metabolites, you know, at some point we're going to have to pick one and move forward with that. Kind of acknowledging that perhaps some metabolites are kind of discriminated for, discriminated against. One of the really notable things from this slide is also that if you look at the green chromatogram, which is the lyophilized sample, those really tall peaks are the short chain fatty acids, which are oftentimes believed to be lost during the lyophilization process. But because the lyophilized samples are powder, they have a greater surface area for extraction of volatile metabolites. And so we're actually increasing the extraction efficiency of them, and you see a increased response. When we look at 4 biological replicates of the lyophilized material, you see that the homogeneity within a storage condition is actually very good. And it indicates to us that the BioCollective is doing a very good job in their method preparation. And so it's feasible for us to move forward with having them prepare the reference material. And so as you can see, I've labeled a couple metabolites that are present in the sample. And you know, one of the things that we really discussed heavily in the workshop is what metabolites should be included in the reference material. And so we came up with a criteria for from the discussion. And really, we felt like metabolites should have health and disease relevance. Marie did a very good job explaining why that is important. We feel that a little less important is that they're part of a biological pathway, because if they are common in a diversity of fecal samples, chances are that down the line, their importance in a biological pathway is still to be determined. Because, once again, we're trying to produce the most scientifically sound reference material we can, we feel like there is some importance that these metabolites should be relatively robust in the measurement science that's involved in this. So we still do want to include analytes that are difficult to challenge, because I think part of the reason why you develop a reference material is to challenge and the variety of analytical methods that are being used by the researchers in the field. And of course, one of the most important things when it comes down to reference material development is the fact that we really need this material to be stable, but not stable over the course of 3 months, but stable on the order of 3 to 5 years. And so that's really important. And from the pilot study we have done last year, we learned some lessons on stability, but of course there's still a lot to be learned moving forward. And so moving forward, I think the choosing what metabolites are going to be certified in the material are based on the expert consensus. So what the both the committee has determined as the most valuable metabolites to have certified. Of course, using the fecal metabolome database that the Wishart Lab has developed is it's just such an invaluable resource. And of course, all of the metabolites that keep coming up in fecal matter through PubMed searches and other available studies. So the table that's kind of pulled out of the paper here shows you some of the top contenders. So of course, short chain fatty acids have very known, one, they're very abundant in all fecal materials, but they also have a number of different biological functions: succinic acid, a variety of lipids, deoxycholic acid, other metabolites that are modified in the liver, neurotransmitters, and then of course the indicators of diet such as enterolactone. And so in the next steps of the reference material development, we are going to be looking at samples from 2 different cohorts, one from an omnivore and one from vegans. We've kind of settled on this is because prior studies show us that the largest difference in fecal metabolites occur due to different diets. This material has already been prepared and it arrived at NIST last week. It is, all aliquots are frozen at -80, some of which are lyophilized, others are just stored in water. One of the things that I did not show you is the fact that when you look at LC-MS-based metabolomics, the preservation buffers create large interferences and oftentimes they just kind of stick to our instruments and there's no kind of work around them. And so we're moving forward looking at samples that are only stored in water. Originally, we had plans to use this material for a long-term stability study. However, due to the current conditions, this material was collected in February and processed in March. And the week it was supposed to arrive at NIST was the week that we got our telework orders, and it spent 3 months sitting in Colorado. And so because we don't have that T-0 time point, we have decided to shift our energy to look at this material more from a research perspective and less so from a long-term stability study. So as I mentioned, there's still a lot of questions that we need to answer before we can fully delve into reference material development. One of which is the effect of freeze-thaw cycles. When the material is collected, it's oftentimes shipped on either dry ice or an ice brick back to the BioCollective. Where it's processed, frozen again, etc. So we really want to see what those freeze-thaw cycles do to the metabolites and bacterial profiles. Also, one of the things that's really important is looking at the microheterogeneity with respect to the subsample size. That's both looking to make sure that the 1 milliliter aliquots that we're currently working with are homogeneous throughout the entire sampling, but also if we were to ship 1 milliliter vials and researchers were only to use 100 microliters at a time, would that homogeneity also stand within that vial and those small aliquots? One of the things that we want to look at is making sure that our extraction methods are actually the best methods to be using for the reference material development. So the pilot study we did last year, we didn't have the liberty of time on our side. And so we just kind of pulled some preps from the literature and went with them. But I think it'd be really valuable to make sure that the methods that we're using are really fit for their purpose. Another question we're trying to answer is that presence of intra versus extracellular metabolites. There is a good chance that storage at -80 is lysing some cells. And then in 2021, we are planning on rolling out a small inter-laboratory study to look at how reproducibility varies across different laboratories. In the metagenomics space, fecal material will never be ground truth in terms of what communities are present. And so one of the things that is being done in that space is looking at organism spike-ins to look at analytical sensitivity and specificity. And so with that, there is no great time to work on a reference material. And one of the things that we want to keep reiterating is the fact that this is a work in progress and it can be iterated on based on what the community needs at the time. So we can expand what metabolites are being measured in the reference material. We can expand in the alignment to health relevance. And as I'd mentioned throughout this talk, this is going to be a reference material, not a certified reference material or a standard reference material, as NIST likes to refer to it, because that is sort of what has been expressed to us from the community. So the major difference between a reference material and a certified reference material is traceability to the SI. And at this point, the community hasn't indicated to us a need to the traceability to the SI. And so right now, we are just exerting our efforts into producing a reference material. But that shift to a certified reference material can happen at any point if the community were to indicate to us that that is necessary. And so with that, I know everybody has said thank you all for your attention. I'd specifically like to acknowledge everyone who helped contribute to the September workshop and all of the manuscript collaborators. Scott Jackson and Catrice Lippa, who are the group leaders at NIST, who are helping this boat stay afloat. David Wishart and Rupa from Alberta, thank you so much for all of your help. And then Raul Kano from the BioCollective. And with that, Marie and I will be happy to answer any questions you might have.